allele. What is the meaning of allele? Alternative forms of a character we call allele. Then in case of blood groups, we say multiple allele. Why? Because in case of Mendel, always we are seeing tall and dwarf are the alleles of a character called height, and yellow and green are the allelic forms of a character called seed color. Round and wrinkle are the allelic forms of a seed shape. Likewise, we are seeing. That means only two alternative forms we are seeing. But here you see, in case of Mendel, that we are seeing more than two allelic forms are present. That's why three allelic forms you can see here. That's why we say this as multiple allele. So that is the first thing you have to remember. Next, the patterns of Mendel, whether it is following or not, that we have to see. What is the pattern? When Mendel said, whenever the two allelic forms of a character are crossed each other, only one form will come for expression. So let us try that. And he called it as law of dominance. When we cross A genotype, homozygous A with homozygous O, we get the children are with heterozygous A. So what we can understand according to this expression is A. So A is the dominant allele and O is the recessive allele. A is the dominant one and O is the recessive one. Next, B. When B crossed with O, so what we get in the offspring children are all with B blood group. So what we can understand, B is the dominant allele and O is the recessive allele. Okay, so in this particular concept, we can say it is following Mendel's law of dominance. Mendel law of dominance, it follows. So first thing is over. Mendel law of dominance. Now the question raises here, whenever A blood group is crossed with B blood group at the time, what happened? A blood group crossed with B, when pure A with pure B homozygous, you have to say. So what happened in first generation, in the offspring, in children, we obtained AB blood group. We obtained AB blood group. So what we can understand according to this, both are dominant. So this is non-Mendelism. So what do you call this feature as? Codominant. This feature can be termed as codominance. So these are the issues related to the blood group. Blood groups are called multiple alleles because more than the two allelic forms are present for a character blood group. That's why we call them as multiple allele. They follow law of dominance. In the case when A cross with O, we get A. When B cross with O, we get B. Okay, so at the time we said law of dominance, it follows. But in one context, that means when A blood group crossed with the B blood group, it is not at all following this particular concept that non-Mendelism we say codominance. Okay, next. This is one of the items. That means issue-wise we are going. So one issue we are seeing here, why they are called multiple allele and why they follow law of dominance and in what case they follow codominance. Okay, next. So how to find out, now here we have to go, how to find out number of genotypes, how to find out number of genotypes based on number of alleles. Now we have to go for that, based on number of alleles. Okay, so for that, let us discuss some aspects here of Mendel again. Whenever the two allelic forms are present, capital T and small t, how many genotypes we obtain? Three genotypes, homozygous tall, heterozygous tall, and homozygous dwarf. In that case, we say there are two alleles and three genotypes. Alleles are two and genotypes are three. So alleles are two alleles are present and number of genotypes are going to be 3. So actually we have a principle for this. What is the principle? Number of genotypes equal to, number of genotypes equal to, I am writing here, number of genotypes equal to n into n plus 1 by 2. The principle here, n into n plus 1 by 2. Okay, so number of genotypes is equal to n into n plus 1 by 2. That we have to remember. N into n plus 1 by 2. Okay. Then let us apply. What is n? N equal to number of alleles. So here you have to remember n equal to number of alleles. Number of alleles. So how many alleles are present in case of the height character, tall allele and dwarf allele. So that's why we say 2 into 2 plus 1 by 2. That
2.
means 6 by 2. That means 3. So total number of genotypes we obtain are 3, and that
3.
is we can see here, that we can see. That means we can apply this principle when we apply in this principle in Mendel taken P-plant characters, then you can see like that. So that two alleles, for two alleles, how many genotypes will come? 3 genotypes. So that is proved here with this here, the principle. Number of genotypes equal to that n into n plus 1 by 2. Now, let us try. Now, let us try the same in case of blood groups also. Blood groups also. Let us try the same in case of blood groups. Okay. How many alleles are present in case of blood groups? I am erasing this. First of all, let me erase this. Now, let's go with the number of genotypes. In case of blood groups, how many alleles are present? There are 3 alleles are present. In case of blood group, number of alleles are 3. What are the 3 alleles? A, B, and O. There are 3 alleles are present. A, B, O. Okay, so for this, what are the genotypes will come? AA, AO, and AB, BB, and BO, OO. These are the genotypes. How many genotypes will come? 6. 6 genotypes will come. We did this all this in case of problems, isn't it? So now let us do the same problem here. Let us do the same problem. I am erasing. Principle is same. Number of genotypes equal to number of alleles. N into n plus that n into n plus 1 by 2. What is n? Number of alleles. So I am applying 3 into 3 plus 1 by 2. That means
2.
12 by 2. That means 6 genotypes. How many genotypes will come? 6. So in case of blood groups, number of alleles are 3 and number of genotypes are 6. Genotype. The 6 genotypes is presented over here. Number of genotypes are 6. Okay. So this is one of the principle about this particular. So finally, let us write what we obtained here. Okay. So in case of blood groups, what we obtained here. Blood group inheritance. Blood group inheritance. So I am asking questions here. Number of alleles. What are the number of alleles here? How many number of alleles are present?
3.
3. Very good. Number of genotypes. Number of genotypes. How many genotypes?
6.
6 genotypes. Number of phenotypes. That means physically how many types of people can be seen over? Think about that. Number of alleles. 3. What are the 3? 3 we have to write. What are the 3? A,
A, O, B.
B, and O. Okay. Next, number of genotypes. We should write AA, AO,
O, A, O, A, B.
BO, AB, O.
A, B.
These are
O.
the genotypes. Okay. Next, number of phenotypes. What do you say? A blood
A,
group.
B,
Generally, when we
W.
see the physical expression we call phenotype, isn't it? A blood group. We say B blood group.
O
Okay.
blood group. O blood group.
We say O
Group.
blood group.
Blood group.
Okay. Next, we say AB blood group.
B blood group.
Yeah.
BB blood group.
No need to say like that. That is genotype. So
Okay.
simply we are saying how many types of people are present by physical expression. So 4 types of people are present. So number of alleles 3, number of genotypes 6, number of phenotypes 4. Okay, for your understanding, in Mendel also I'll do the same. That in case of pea plant, number of alleles 2. What are the 2 alleles? Capital T, small t. Okay, number of genotypes 3. Homozygous tall, heterozygous tall, and homozygous tall, heterozygous tall, and homozygous.
Homozygous dwarf.
So that means three. Okay. Number of phenotypes, only two, tall plant and dwarf plant.
Dwarf plant.
So that's it. There are two phenotypes, three genotypes, two alleles. So blood groups, three alleles are present, A, B, O. The six genotypes are present, A, O, B, B, O, and A, B, O. Next, there are four phenotypes are present. Okay. So this is one of the issue. And issue wise we are going here. Issue wise we are going. So one of the issue we are completed. Okay. Next, let me talk about some other issues here related to the blood group inheritance. Here, we need to know what it is exactly. So that's for that I am drawing four RBC here. Four RBC. Four types of RBC in four kind of people. Biconcave shaped RBC. So scientists have noticed that in case this in some RBC, they identified a sugar polymer. Okay, in that kind of sugar polymer, they get identified, they get noticed a sugar polymer, and another type of sugar polymer noticed on surface of RBC, Donan's membrane, we use it to say. And in some people, they noticed that both types of sugar polymers are present. So I see here, I'm not using antigen, antibody, these kind of issues here. Just the plain topic I'm talking. Some of the RBC is not at all having any sugar polymer. So they named it as that particular sugar polymer, one as A, one as B, and here it is A and B again. Okay, so those who don't have any these kind of sugar polymer, they said no sugar polymer is present. You see, I'm using again and again the word sugar polymer. That was the 2020 Neet question. So that means people believe so that is antigen. Actually, if those who are having as an antigen concept, they'll be confused over when it is asked over. Okay, so when you call it as antigen, that you should know when we call this as an antigen. So these are the four types of 780 crores of population in the world. But we can classify them based on their blood groups as four types. Some have A sugar polymer on the surface, some have B sugar polymer on the surface, some have both, some have nothing. We named it as, we named them as A blood group persons, B blood group persons, and both sugar polymers, if they are present, we say AB blood group person and O blood group person. We need to use that word antigen, the word very carefully here. So there are four types of blood group, phenotypically, four types of blood group. When you write their genotype, that means to get these particular, there are two possibilities. I am writing genotypes here even, like AA, homozygous A or heterozygous A, homozygous B or heterozygous B, then AB, then O. So these are the possible genotypes in this particular case. There are two genotypes possible for A blood group and two for B blood group, one for AB and one for O. So that means here if I am A blood group person, I am having A blood group, means that you need to understand, and on my RBC there is a sugar polymer called A, and that my genotype may be AA or AO. My genotype may be AA or AO. If I am the B blood group person, on my RBC there is a sugar polymer called B, and my genotype may be on chromosomes you'll find, isn't it? On chromosome, ninth chromosome of the human being is having. So on my ninth chromosome, either BB or BO, homozygous B or heterozygous B can be seen out. If I am AB blood group person, if I am the person with AB blood group, I have both A sugar polymer and B sugar polymer. Both are present and my genotype is one type, that is AB. So some RBC will be like this, like O. That means no A sugar polymer, no B sugar polymer. Okay. So when does it is called antigen? That is very important here. For that, If you know any basics of blood group, just please forget about them. Then you'll understand why it is, we use a word called antigen. So we know actually the definition for antigen. We completed that in immunology. We say, what is antigen? People say antigen means foreign body. Antigen means foreign body. Okay, foreign body. That means a bacteria can be an antigen, Salmonella typha, and a virus can be HIV virus, like coronavirus, whatever. All we say that antigen, isn't it? Foreign body is called as antigen, no doubt about that. So when why that word A B as an antigen? That we are trying to understand now. That we are trying to understand now. Listen carefully. So when the, for example, if I am taking O blood group, a person O blood group means immediately you need to understand on the surface of that person's RBC, either you don't find A antigen or sorry, A polymer or B polymer. Now all the remaining people are there in front of this particular person. I am writing that A blood group person, B blood group person, AB blood group person. Let me say once again that A blood group person is having A sugar polymer, and B blood group person is having B sugar polymer, is having B sugar polymer. Let me change the color, use blue. B sugar polymer, and the AB blood group person is having both A sugar polymer and B sugar polymer. These are the cases. Okay. So what next? What you don't know, that is the antigen, foreign body. In general we say the foreign body concept. That means anything if you don't know, anything if you don't know, that we consider it as antigen. The thing which we don't know, we consider as antigen. That is very important. Okay, the thing which we don't know is considered as antigen. Okay, then who is antigen? Now let's talk about that word antigen. For example, if this particular A blood group person RBC enters into here, what do you say it as? So for O blood group person, for this person, A is antigen because we don't know that. A is antigen. Okay, is it erased? Okay, is it erased? Right. Like, let me write here. This is B, and okay. So then next, let's talk about this. A is the antigen for him. Okay. Next, for this particular person, B blood group containing person. B is the antigen to O blood group person. AB is the antigen to O blood group person. So what I am talking here, when we say antigen, when we don't have, we consider it as antigen. When we don't have, it can be considered as antigen. That is very important for us. So that is very, very important. Okay, so let me say once again, a person, O blood group person is there. What he know? He don't know anything. He don't know A sugar polymer and he don't know B sugar polymer. So that's why for him, A is the antigen, and here it is the A blood group person. Let me write here. It is the A blood group person. He is A blood group person which contains A sugar polymer. Okay, next, B blood group person. B blood group person. So that he is the AB blood group person. He is having both B blood group, AB blood group person. He is having both. Okay. So what do you say it as? A blood group, B blood group, and AB blood group. All the three for him antigen. So let us write notes for this. Number one, for A is antigen for O blood group person. A is antigen. A blood group contain A sugar polymer. B is antigen. For him, A is antigen, B is antigen. So that's why what he tried to do, he will try to produce antibodies against them. That's why in his plasma, if you inject only, if you inject, he will try to produce antibodies, A antibody and B antibody. Okay, so finally we can declare like this. This is a completely new concept, not like you learned earlier. So that's why that till the time I used a word sugar polymer, now I am using this word antigen. So A and B both are antigen to whom? A and B both are antigen to this particular person, O blood group person. Okay, so that's why I'm writing here O blood group person. No antigens on surface of RBC. No antigens on surface of RBC. The first thing you have to remember, on his surface of RBC, there is no A antigen and there is no B antigen. Okay, no antigens on the surface of RBC. Then what about the antibodies? He is having A and B. Both antibodies are present in the plasma because when he exposed to such kind of blood, that he able to produce both antibodies, AB antibodies on the surface of plasma. On the surface of plasma. Okay. So on the surface of plasma. So that is in case of O blood group. Okay. Now let us try about the A blood group person. About the A blood group person. So now we are going with A blood group person here. In case of A blood group person. So what is the meaning of A blood group? He is having on his surface of biconcave shaped RBC. So he will have A antigen. He is having A antigen. We can't say antigen actually. So when we say antigen, here it is. So for him it is not antigen. Okay, he is having A sugar polymer on the surface of RBC. For him it is a sugar polymer. Now let us draw that the the four types of RBC again here. Four types of RBC I am writing here. Four types of RBC. Now let me see whether you able to say or not. So I am naming here. This is A blood group. This person is A blood group, and this person is with B blood group, and this person is with AB blood group, and this person is with O blood group. Okay. Now I declare their blood groups. Okay. So let me put all of them in the box. So boxes I'm making here. So now you locate first of all their antigen. That means the so-called sugar polymer. What about an A blood group person? What sugar polymer is present?
A.
A. Very good. A sugar polymer is present. Now, what about an B blood group person? What sugar polymer is present?
B.
B sugar polymer. Okay. Next, what about AB blood group person?
AB.
Yeah, he is having both A sugar polymer and B sugar polymer.
B sugar polymer.
What about O blood group person?
O.
Nothing, nothing. That means he don't have A or B. You see the RBC. That's why I'm showing with visual. Okay, nothing. Don't say O. O means zero. Okay, so
Okay.
now let me give the blood here. A blood group to A blood group person. Did anything happen? A blood group to A blood group person. Anything is happened? Can you consider as a antigen? No. Whatever you
Okay.
have, the same you are getting. So that's why you don't feel it as a antigen. Okay. Next. So B blood group person has given blood to him.
Yes. AB.
Ain't AB? B blood group person is given blood to him. Okay. B blood group person is given blood to him. What happens? I am asking. That he don't know
Antigen.
B. He don't know B. That's why he consider B as an antigen.
Antigen.
Atla. He consider B as an antigen. If AB blood group person is given blood to A, so he consider B as an
Antigen.
antigen, like that. So O, when O blood group person is given, any problem? No, not at
No.
all. Why? Because he don't have anything. That O blood group person is not having anything. Okay, so when we consider the antigen, if I am A blood group person, if I am the A blood group person, I consider B as an antigen. If I am the A blood group person, I don't consider A as an antigen because what I have, if you have the same, no, I don't consider it as antigen. That is the antigen concept. O blood group don't have any sugar polymer. That's why giving blood O is not at all problem to any time. So let me write here for the similar kind that AB blood group. Let us write for the AB blood group here. That AB blood group. I'm going to write AB blood group here. So what is the type of RBC in AB blood group person? What type of RBC can be seen over? What he have on the surface? On his surface, what can be seen over? He is having A antigen, A sugar polymer, and he is having B sugar polymer on his surface. So that's why he consider as AB. That's why he consider as AB blood group person. So that you have to remember a person with AB blood group is having both A and B. Both sugar polymers are present. Now let me introduce you the four types of people in that world, the four types of people with their four RBC. So one type of RBC, other type of RBC. So I am drawing that four types of RBC. Don't look toward the shape. Yeah. So some people are having some A sugar polymer on their surface. What do you call them as? Some people are having A sugar polymer on their surface. What do you call them as? I'm asking you.
A.
A blood group. We need to say it as A blood group. Some are having B sugar polymer on their surface. What do you call them?
B blood
B
group.
blood group.
Yeah, very good. Some are having both A and B sugar polymers on their surface. What do you call them as?
AB
AB
blood
blood
group.
group.
Yeah, very good. AB blood group. Okay. Next, some don't have any kind of sugar polymer.
O
O
blood
blood
group.
group.
We say them as O blood group. Let me fill them in the boxes before we identify them. Let me fill them. So four types of people. Now, four types of people. So how do I respond with them? I am A blood group person now. Here in the box, I am A blood group person. So how do I respond against them? That I have to see. Okay, how do I respond? First of all, okay, so first of all, let me announce here. What is this? What do you say? This is A. And what is this? This is what blood group person he is?
B blood group.
A, because he is having B sugar. And what about him? AB.
AB
AB.
blood group.
Yeah, he don't have any sugar polymer. What about him?
O blood group.
Okay. Now let's make it individual person. That means these four person came to give blood to him. All the four persons came and to give blood to him. So anything happens. That means if you are the person here. Let me write. Let me name his name as Srikanth. So I am Srikanth. So my blood group is AB. Okay. So A blood group person came to me and given blood to me. Okay. So do I feel anything antigen?
No.
No. Why? Because I have the similar kind. You see here, I have the similar kind. Meedityanale, Madityanale. Matching. Next, B blood group person.
No. Same unnai.
Same. And similar kind I have. Okay.
Yes.
Similar kind I have. So that's why I don't consider that also. Okay. A B blood group person.
No.
No.
Same unnai.
Yeah.
Similar.
That means what I have, if you give the same, I don't feel. That means So for AB blood group person, who is the antigen?
O.
O blood group. O blood group.
ఓ అంటే ఏమీ లేదండి. యూ రిమెంబర్ ఇట్. అందుకే మీ ఓల్డ్ నాలెడ్జ్ ఓవర్కమ్ చేస్తూ ఉంటుంది. నేను ఇప్పటికే 3 టైమ్స్ చెప్పా. ఓ అంటే ఏమీ లేకపోవడమే ఓ. ఓ అంటే 0. ఓకే. సో అంటే ఇక్కడ ఉన్నవి రెండే. ఓన్లీ 2 sugar polymers are there. A and B. From the beginning we are focusing on them only. A and B. O means nothing. There is 0. Okay, now let's come with the O. O blood group. What they have? An RBC. ఏముంది RBC మీద?
No antigen.
Huh? On the surface of RBC, what is present? On O blood group, surface of RBC contain what?
No antigens on surface of
Yeah,
RBC.
there is no antigen. Very good. Now, if A blood group person blood is injected to the O, what happens? Whom he consider antigen? He considers
A
an
antigen.
antigen. For B blood group person, he don't know anything. He don't know anything. That's why A is antigen to him. B is also antigen to him. Now they see the most contrast issue here. That B blood group, A antigen, B, both is having. So that's why he don't consider it as anyone as an antigen. That's why we say no antibodies. He is not able to produce any antibodies. He is not at all able to produce any antibody. That you have to remember. Okay, is not at all able to produce any antibody. Okay, next. Next, here the last one we have to go with okay, B blood group. The B also the similar. Similar, that means what we have to say. Let me erase some issues here. There you can understand. In case of B blood group person, what is absent, what is present. So that you have to see. What is absent, what is present. Means I have to draw that particular B blood group. No need to see here. We know already. So is the B blood group person. Okay, I am the B now. So if I am the B, so what can be expected? You just imagine it. No need to explain. You just imagine it. I am the B blood group person. You just